Okay, and I think we have a couple more people coming in right now. Yeah. All right, yes, and we do have everyone. So I will go ahead and start by saying welcome. My name is Haley Callison. I'm the Biocontainment Institute Administrator. So we are very excited to have all of you here. This is our inaugural training event. So we are very, very happy and excited to have this group with us today. I'll just start with saying that this session is a testament to the dedication and expertise of our teams. They play a pivotal role in advancing the safety at the university, but also on a grander scale across the world. So we're incredibly proud of our program. We've experienced a huge amount of growth even within, which attests to the expansion and the need of biosafety and quality assurance for our labs. And we provide these services across 3 campuses. At our heart, we are a service department, so a lot of what we do, our biosafety team is in constant collaboration with our primary investigators, our facility staff, just to ensure that everyone is fostering a culture of safety throughout the university. Notably, our biosafety team has kept our biocontainment lab very, very safe the past few years. And also we do have staff that has been on the forefront of securing a polio essential facilities designation for the university. With that designation, our quality assurance team has been right there with them, supporting, and they've been instrumental in laying the groundwork for that program. But then also our quality team, they have had lots of success on their own. They were at the forefront of developing the coordinating center database, which all of our NPRCs stateside are using to house their COVID-19 data. And that's a project that's hugely significant just because of the importance that it plays in the future pandemic responses. Additionally, we do have our Select Agent program at the Primate Center, which again has been a huge benefit, and our team is there working with our high containment performance research board to ensure that our studies are done meticulously, the planning, the prioritizing, the timelines, and the rigor to minimize risk and cost are all in place. And we're hoping to share this with you today just to show how all of those pieces and parts and all of those different disciplines work together to advance our posture so we can have a very quick turnaround whenever there is an emergency to respond to. So now that I've kind of spouted on about our staff, I'm very proud of them. I will ask and reiterate that if you do have questions during the presentations, to please add those to the chat. We will go back to them and have time for everybody to discuss and kind of delve deeper into each of the sessions with our speakers. And I think that that's all I have. Thank you for listening to me ramble. And from here, I'll go ahead and turn it over to Tammy BAVARET. She will be our first session speaker speaking on biosafety program fundamentals.
Thanks, Haley. So before I jump into the content, I wanted to give a brief moment to ask you guys if you had any pressing ideas that when you saw this title and you signed up for it, was there something that you were thinking you wanted to learn or that you were hoping to hear about? And if you could just, either drop them out in the chat real quick or come off mute and just, state that, we'll make note of it and we'll see if we can address that as we move through the presentation. Okay, so without further ado, all right. So again, my name is Tammy BAVARET. I am one of the assistant directors for biosafety at Tulane University. Thank you again for joining us for our first Tulane University Biocontainment Institute's Building Blocks for Success, Creating a Strong Foundation for Your BSL3 Biocontainment Program. And to get us started off today, I will be introducing the fundamentals necessary for building a biosafety risk management program. with a highlighted focus on BSL3 best practices. But before I dive in, I wanted to kind of give you just a quick outline of the topics that I'm going to be covering today. First, we're going to, of course, do a brief overview of basic biosafety principles. You can't have a biosafety talk without that. And I'll highlight some key features of BSL3 that we'll be covering in more detail later on in a later session. I'll introduce you to a framework for biosafety programs that's fairly new, ISO 35001. This is in addition to the CDC's Biosafety and Microbiology and Biomedical Laboratories manual that we pretty much consider our biosafety Bible. But this is fairly new and it's very important. So that's going to be a good chunk of the talk today too. And then we'll talk about considerations for building the infrastructure of your biosafety program. And lastly, we'll discuss developing and implementing biosafety procedures, where we'll cover things like training and competency assessments, emergency preparedness and response training, considerations for facility design and maintenance, documentation, record keeping, the importance of fostering a culture of safety, all in order to maintain your biosafety program for BSL3 work. So at its heart, all laboratories have occupational hazards. This may not be a comprehensive list, as it is, of course, it depends on the activities being performed in the laboratory, but generally most labs have these common hazards present. So when we narrow down and focus in on biological or microbiological laboratories, we are now faced with additional hazards. The biological agents themselves and the biological materials worked with in these labs present the possibility of causing harm to the people that are working with them, potentially leading to laboratory-acquired infections. Hence the term biohazards, which are biological organisms or their products that can cause harm whenever, whether it be a disease or toxicity, or induced unwanted mutations in humans, animals, or the environment. Specific examples of these include things like infectious agents, pathogenic bacteria or viruses, toxins of biological origin, recombinant or synthetic nucleic acid molecules, including viral vector systems such as adenoviruses, adenoviral vectors, lentiviral vector systems, or non-viral vector systems such as those that utilize CRISPR-Cas9. And then anything that may have come in contact or be contaminated with those above materials are considered a biohazard. So now, the definition of biosafety is simply put, the practices that reduce exposure and release of biohazards. So when we're going to talk about biosafety level 3, keep in mind that all biosafety practices are based on a risk assessment. It's not really one size fits all. But what does that mean? So generally, a risk assessment is the process utilized to determine what the risk of a particular activity is. Specifically, a biohazard risk assessment assesses the biological agent's characteristics, the harm it causes, any susceptible hosts, what hazards are present based on the specific activities of that laboratory and what they wish to perform with the agent. Here are just a list of some common factors that we consider when we want to perform a risk assessment. These are definitely not everything, but it's the top. Things like, what's the pathogenicity? What is the route of transmission? How long is the agent stable in the condition that it's used in? How it's stored, if it's stable, and how it's stored. How stable is it if it ends up on a surface and it's not disinfected or mist? What is the infectious dose? How much of it will be worked with at what time? I'm relating that to the infectious dose. What's the experience level of the workers? Do they need training? And then how will medical surveillance be conducted? Do you have a medical surveillance program in your institution? And so when considering these things, a risk assessment is not linear. It's an ongoing process. It doesn't just occur one time. And so it involves first identifying what the hazard is, and then identifying who it affects, who's performing the activity, but also who might be affected should an incident occur. will it affect neighboring or outside or, you know, beyond the person actually handling the material. And then evaluating the risk level. So if there were an incident, how serious would the consequences be? And then how likely or how probable is that incident to occur based on what the activity is and where it's being performed, that sort of thing. And then determining if there are protections that can be put in place that would minimize or prevent an incident from occurring, or that would lessen the consequence if it were to occur. And so this could be things like specialized PPE, adjusting your facility requirements, recommending vaccines, and such. And then once that's done, it's important to document the determined risks and then and any mitigation plans that will be implemented, and then follow-ups to determine how well it's working, or if improvements or adjustments need to be made. And this comes from feedback through inspections with the laboratory personnel. And so as you can imagine, biorisk assessments can be very complex and require the input and feedback from several subject matter experts. So it is not done by one person, or it's definitely not done alone. Commonly PIs or the investigators themselves and biosafety officers can perform these assessments together. But as your program grows, utilization of an institutional biosafety committee can make the process much more streamlined, consistent, and effective. And later we're going to be discussing some of the components of setting up an IBC. So remember, the core principles of biosafety are to protect personnel, the environment, and the public from biological hazards. This involves implementing safety protocols and conducting regular risk assessments. So what are some of the ways we mitigate biorisk and ensure that containment is maintained? You'll hear a little bit more about this later in Anne-Marie's talk, but at its outermost level, the facility acts as a secondary barrier. The design and construction contribute to environmental protection. And then within the lab, safety equipment and personnel protective equipment act as the primary barrier between the worker and the agents they're handling. But perhaps the most critical is the training and expertise of the laboratory workers themselves, because strict adherence to safety practices is ultimately what minimizes the greatest risk. So now we're going to move on to a recap of what biosafety levels are, just as a little refresher. Biosafety levels correspond to the facility design, the protective equipment, and the laboratory practices required to handle the specific agent safely. And so each biosafety level builds upon the foundation of the previous one, adding safeguards as the risk increases. So biosafety level 1 is generally appropriate for those agents that do not cause disease in healthy individuals. Biosafety level 2 is appropriate for work with agents that cause disease, however, it's typically treatable, has maybe a low probability of occurring. And then biosafety level 3 is used for work with agents that are associated with serious or life-threatening disease that has the potential for respiratory transmission. And biosafety level 4 is reserved for those exotic agents that pose a high risk of life-threatening disease and is transmitted via aerosol and also has no vaccine or therapy available. So this course focuses on BSL3 labs, which are applicable to clinical, diagnostic, teaching, research, or production facilities where work is performed with agents that may cause serious or potentially lethal disease through inhalation to their personnel and may contaminate the environment. And so the key safety measures in BSL3 labs, which are going to be going over in much more detail a little later, just touched on here, include things like controlled access, specialized ventilation systems, biosafety cabinets, personal protective equipment, and these measures ensure that researchers are protected from exposure to hazardous agents. Some special features may only be required depending on the findings of the risk assessment that you perform. And these are highlighted in green in this particular image. You may not always need an effluent decontamination system. You may not always need a shower out. It depends on on the actual work that's being done itself. And so, for example, Our BSL3 labs are equipped with HEPA-filtered ventilation systems that prevent any airborne pathogens from escaping the lab environment, and we have the capability of isolating rooms for effective decontamination. We also have some labs, we have some facilities, we have a facility that has a shower out requirement based on the work that's being done, and we have another one that doesn't require a shower out based on the work that's being done. And so we're going to move into sort of the framework for biosafety programs. ISO 35001 was fairly recently published. It provides a framework for managing risks associated with hazardous biological materials. It is an international standard that outlines best practices for biosafety management. And by adopting this ISO 35001 at Tulane, it has helped us standardize our biosafety practices and ensure compliance with both our local, national, and international guidelines. And so the key elements of ISO 35001 include risk identification, assessment, control, and monitoring. And by following this framework, we can ensure that our biosafety programs are comprehensive and effective. And the following topics that I'm going to discuss throughout the rest of this talk are laid out in ISO 35001, and we're going to go over the key takeaways from these components that are a best practice when building your biosafety program. And so the first is stakeholder relationships. It should be intuitive. Building relationships with your institutional stakeholders is critical for gaining the needed support and resources necessary to implement your biosafety program and mitigate risk perception and foster a culture of safety. So this includes collaborating with institutional leadership, researchers, administrators, and your safety officers. We regularly engage with our stakeholders. We conduct workshops and meetings to ensure that everyone is aligned on biosafety priorities for the institution. And so the next is potentially considering setting up an institutional biosafety committee. And while ISO 35001 refers to an institutional biosafety committee or an IBC as a biorisk management committee, regardless of the name, these committees play a vital role in overseeing research involving hazardous biological materials. They review research proposals, conduct risk assessments, and ensure compliance with biosafety regulations. It's important to establish a charter or functional role for this committee with the buy-in of your leadership so that the committee has the authority to implement and enforce the institutional biosafety policies. Our IBC meets monthly to review new research, discuss any biosafety concerns, and this collaborative approach helps us address potential risks early on before the work is started. So when setting up your IBC, the committee should consist of members who are independent of activities being reviewed for biorisk issues. And the members should be diverse with enough expertise in the relevant areas that will be possibly reviewed for effective biorisk reviews. Now, obviously these are your researchers possibly serving on this committee, so you're going to have conflicts of interest, and so there needs to be a mechanism for them to excuse themselves and then come back in and be able to discuss. So that's laid out in the ISO as well. A chair needs to be appointed by senior leadership to facilitate the meetings and provide reports back to the regulators and senior leadership if required. But depending on the size of your institution and program, it may be advantageous to consider having a dedicated administrative support to run the IBC. This would be for things like collecting registrations, assisting the researchers with edits, modifications, generating agendas, minutes, publishing policies, submitting regulatory reports, and just general coordination of scheduling the meeting. Assessing if this is a resource that you will need should be considered and communicated to your stakeholders early on in the process so that you can advocate for this resource. You can always go in though and justify why you need it now as you grow though too. So the ISO refers to a biosafety officer as a dedicated a biorisk management advisor. We call them biosafety officers for the most common part. This is essential for planning and implementing and managing the biosafety program. The BSO ensures compliance with regulations and provides guidance on biosafety practices. Depending on the size of your program, you might need to consider the need for assistant biosafety officers in order to reach all of your laboratories. And the exact title that they're given is not really important. It's more about the function that they provide. So once you have your IBC and your biosafety program established, it should be utilized to perform regular risk assessments to help identify new risks and ensure that existing controls are effective. So as mentioned before, this process involves evaluating potential hazards and implementing mitigation strategies. So, for example, we conduct annual assessments on all of our labs with biological hazards. So that's once a year, which includes reviewing what is registered with the IBC, their safety protocols, inspecting the equipment that they use to make sure it's certified, and interviewing lab personnel to make sure that what they say they're doing matches what they're approved to do. And then our IBC administration does post-approval monitoring 18 months post-approval, and renewals are required every 3 years to ensure that we're always aware of any changes to research projects that inevitably occur. That's not mandated anywhere. You have the choice of doing it annually, 3 years. You can set your own sequence based on the needs of your institution. So while specific research conducted in laboratories can vary greatly, there are many general safety practices that can be standardized across all labs. Standard operating procedures, or SOPs, provide clear instructions for all biosafety related activities. They ensure consistency in safety practices and should be regularly reviewed and updated. Biosafety program SOPs, for example, can include guidelines for handling emerging pathogens, biological spill cleanup, and emergency response procedures. So once you've developed your biosafety program, IBC, and the institutional policies and SOPs, it's important to effectively communicate this information to your research community. Comprehensive training programs are essential for ensuring that all personnel are knowledgeable about the biosafety principles, emergency procedures, and the proper use of the safety equipment. Quarterly training sessions and lunch and learns are one good way to keep staff updated on the latest biosafety protocols. And these sessions can include hands-on training, interactive workshops. Just try to keep it fun and bolster engagement and participation. Particularly important in high containment laboratories or BSL3 laboratories is regular competency assessments to help identify areas where additional training or procedure adjustments are needed. These assessments ensure that all staff members are proficient and continue to follow the required biosafety practices. An additional critical topic to cover in your comprehensive training program is developing and communicating clear emergency response plans for handling incidences such as spills, exposures, equipment failures, etc. Our emergency plan, for instance, at Tulane includes detailed procedures for responding to various biosafety incidences such as spills, pathogen exposures, weather events, man down scenarios, and insider threats. And then another great way to test the efficacy of your training program and reinforce what is taught is conducting regular drills. So this helps ensure that staff are prepared for various emergencies. These drills also help identify gaps in emergency plans and improve response times. And so I highly recommend involving your outside emergency responders in on your drills, as it's an excellent way to test your plans and ensure what you think will work will actually work, and ensures that the local emergency responders know what they should be doing when they respond on site. So we'll just touch on a few design considerations, but we'll dive into it a little deeper later. I just wanted to highlight that it's important to design facilities with biosafety in mind, to include features like controlled access, ventilation systems, decontamination areas, because proper design is essential for maintaining a safe working environment. And then regular maintenance of biosafety equipment and facilities ensures that all safety systems are functioning correctly. This includes inspections, equipment, maintenance, and upgrades. We do have a dedicated maintenance team that conducts regular inspections of our biosafety cabinets and ventilation systems to ensure that they're in optimal condition. So if you haven't heard the quote, maybe you have. If it's not written, it never happened. And from a regulatory perspective, that's absolutely true. Maintaining detailed records of all biosafety activities is essential for demonstrating compliance and assuring accountability. Records include things like training logs, risk assessments, incident reports, maintenance logs. Basically, everything that we've talked about needs to be documented so far. And so there needs to be a plan for that volume, for what you're going to do with those records, what systems you're going to use to maintain these records, as well as the security that may be required to keep them, especially if you're going to move to a paper-free environment, electronic-based systems. So cybersecurity is important. And then depending on your local and government regulations for the field that you're in, you'll very likely be subjected to a regulatory audit at some point in your future. And regulatory audits help identify areas for improvement and ensure regulatory compliance. And of course, documentation is crucial for demonstrating compliance during these audits. They're going to ask for those documents that I mentioned before. And so conducting self-audits ensures that you're always audit ready. Now, moving on to fostering a culture of safety. It really does start at the top. Leadership plays a vital role in promoting a culture of safety. Leaders should actively support and participate in the biosafety initiatives. At Tulane, our leadership team regularly participates in biosafety meetings and training sessions and are invited to safety drills in order for them to set an example for the rest of their staff. And then the next is, of course, staff engagement. So encouraging staff to take ownership of the biosafety practices and having them give input on what they need to do fosters a culture of safety. Engaged staff are more likely to follow safety protocols and report potential issues. So at Tulane, we have a departmental safety representative program where staff members are recognized for their commitment to lab safety, and these individuals are responsible for ensuring that their department is following the safety practices that the university has in place. And this program has significantly increased staff engagement and adherence to the safety practices or protocols. So coming up at 1:30, just to kind of recap in summary, setting up and maintaining a robust biosafety program involves understanding BSL3 requirements, implementing a comprehensive biosafety framework, developing and maintaining the infrastructure, and fostering a culture of safety. So I encourage all of you to take these principles back to your institution and work towards strengthening your biosafety programs. Thank you again for your attention, and I'm happy to answer any questions that you may have.
Okay, so we don't have any questions in the chat at the moment, but I would encourage everybody, if you do have questions, that you do have a small break, you can just open conversation. So feel free to engage.
I'm having trouble hearing you, Haley, sorry. So I do see some questions in the chat. Let me see. Let me scroll up and kind of catch the beginning.
Okay, so yes. You want me to go ahead and post it, and we can start on. So the first one is from Luis, and says, what are your comments regarding the elimination of RG from BSL and the new WHO LDM for the business? for something that will impact the risk perception at UA.
So it's a great question, Louis. Here specifically in the US, I don't feel like it's going to affect us that much because we are still having to follow the BNBL. But essentially, it's probably going to eliminate a little bit of the confusion between risk groups and biosafety levels. So, you know, there was the 4 risk groups that were kind of spelled out the same way, you know, causes disease, doesn't cause disease, causes severe disease, you know, for risk groups. And then the biosafety levels very closely correlated, but then you didn't always have the risk group 3 agent at BSL3. It depended on its route and what the work being done was actually. So it might actually, in my opinion, this is just my personal opinion, it might actually simplify the confusion in the long run by just referring to biosafety levels. You're welcome. And then I see there's another one. Let's see. Okay, so I'll just read it out loud since I'm looking at it. So Louis also asked, I know competency assessments will depend on a case-by-case basis. Do you have a minimum of time required by users for having full access to work at the BSL3 spaces? So I can answer some of that, but I don't know if Anne-Marie would like to come off and talk more about the competency training, both the startup and the continuing.
Sure, thank you, Tammy. For here at Tulane, we do proficiency-based training. It's not based on time. It's based on procedures done within the laboratory. So we've established a check sheet of procedures that we feel trainees need to know prior to going into the BSL3 for full access on their own. So we do proficiency-based, and then we check that proficiency through our office by doing final walkthroughs to verify that they understand the safety procedure practices and even incident response and emergency preparedness within the laboratory prior to getting access to the space. So it is very dependent on the person, and that is what we tell our staff. It is not it's not time. One person could take a month, another person could take a couple of weeks. It's very dependent on how much time they get in the laboratory, their experience and comfort, and understanding the practices and procedures.
May I ask a follow-up question associated with that? This is Alicia.
Sure.
So where I work, we have a variety of individuals who do a variety of different jobs, and some of them are like techs, some people manage inventory, some people are researchers who work with biologics, some are only working with chemistry. But we're all kind of in areas where we all have access to them. It's just, you know, don't go into this door if you're not part of this department. So it's not really cordoned off security-wise. Would you recommend that under BSL3 circumstances, if you're not actively working with that agent, does everyone need to get trained? Is like hearing just the risks associated with that area and making that information available, but not doing hands-on training enough? Does everybody need to like see, you know, this is why this is important. Here's the safety measures that we have in place. You know, how much do you train people who are not directly going to be interacting with BSL3 risks? I guess is my question.
Yeah. So there's two kind of scenarios going on here. One is the select agent space. So it's very restricted. They don't have access to the space that they're not working in. So they are trained specifically. But to answer your question more general for what we do on in our other lab that's not select agent, it is something that needs to be looked at. What are they doing? You know, do they need to be aware? Depends on what they have access to. So if in your scenario, everyone has access to everything, I would recommend that there is at least we call them kickoffs, but I do recommend that there's at least a team meeting training where they're aware of what's being worked with, what their risks are. You kind of do a topical agent training, going over what's going to be worked with and what they're allowed to do and what they're not allowed to do. If you're tying it back to, do they, they're just now onboarding and do they need to have all of this extra training? No, they only need training for the job they're going to perform, but they do need to know what they're allowed to then access once they're given access. And it does become a trust thing, but the people that are in the lab working in there will also should have a good relationship with you to report back when someone's, maybe doing something they're not supposed to be doing. And then I kind of am I may be leading in or assuming, but I could see you may have an issue with who trains them, because everybody's doing a different job. So who trains who? In our case, we typically have like a senior person take over and kind of train the trainer. This person takes them on as a mentor, like they're the mentor and they're menteeing a newer employee that's joining, and they're the ones that are teaching them and training them on that specific activity that they need to perform, and then is signing off that they have done it proficiently a certain number of times, and then they report that back to the biosafety officer, and then the biosafety officer can come in and observe them do it to confirm proficiency if we felt that was needed. And so that's one way that we approach it too. So I don't know, that's kind of a lot. I hope that answered your question. If there's more to it, feel free to clarify and I can provide more.
That was great. Thank you so much.
Okay. Well, I can stop sharing my screen and we can move on to the next session if you're ready.
All right, so for a slightly different topic here, we're going to be talking about quality management systems and the importance of having a quality management system to actually have a robust biocontainment program. So many of your biocontainment programs may not have a requirement to have any sort of official quality assurance regulated environment, such as you may not be doing GLP studies or GMP testing, but I'm going to talk to you guys about why quality practices are a benefit to any biocontainment laboratory that you might be working with. So very quickly, what we're going to go through are how you start thinking about making a quality assurance unit, because if you haven't had a requirement for one for any regulatory reasons, where you might want to put one, what you might like the scope of that unit to be. And then we'll talk a little bit about the best way to actually design and implement one of these quality management systems. But if you take nothing away from the next 20-25 minutes, the most important thing if you're looking to build a biocontainment program is that if you want quality within your biocontainment program, it's going to cost. It's going to cost resources, it's going to cost time, and most importantly, if you don't have any existing quality systems, it is going to require cultural change. But the best way to mitigate those costs of building your quality within your biocontainment program is to use a risk-based approach. And what we're going to talk about today is how we can build a quality management system with that risk-based approach to really have an efficient quality system that hopefully is not viewed by those participating in it as red tape, because oftentimes quality assurance programs can be seen as red tape to hit regulatory matters rather than as something which benefits the biocontainment operations as a whole. So I mentioned, how do you go about starting up your unit? Well, there are a couple of key questions that you have to ask. And the key questions are absolutely critical, because without doing these prior to establishing your unit's not going to know what areas they provide oversight for. So the reason I say this is with a biosafety unit, right? It's very simple. If it's a biological risk, then it's controlled by the biosafety unit. Every single process in your institution can have a quality management system requirement for it. So if you're establishing a quality assurance unit, what are they overseeing? Are they overseeing your compliance programs? Are they overseeing your research programs? Are they overseeing your facilities and your operations? If you don't scope out where your unit's responsibility is prior to even developing the unit, you can't build it in a way that actually is efficient for the system that you want to assess. And then in line with the scope, you need to know the purpose. What are your requirements? So if you have GLP studies, if you have GMP studies, then obviously your requirements are going to be based upon the regulatory requirements of those systems. But what above and beyond the regulatory of the requirements of those systems do you want in your program to ensure that you are actually able to do the work in a way that's high quality and, most importantly, is safe? So as you're scoping out your quality assurance program, once you know what the scope is and what they oversee, you need to appropriately place them within your organization. And this may seem like an obvious thing, but it's really, really important to ensure that your reporting structure for your quality assurance unit is effective for the scope of the work that they're doing. So it doesn't matter how you have it set up. In this basic example here, we're looking at a biocontainment facility, right, which has some level of top management. This is your executive director management, your top management. All of these are placed at the top of the scheme. And however your quality assurance unit reports, they have to report separately from the people who are operationally doing the processes. So you can't have a quality assurance person reporting to a lab person. You can't have a quality assurance person reporting to a biosafety officer, right? You have to have a good, strong top management where all of the reporting for your quality systems are going through the same, it can be the same person, but are not going through someone who's operationally managing and running those individual programs. So once you've established, and the reason I want to emphasize this is I just mentioned quality assurance can do a lot of different things. So for example, there's quality assurance within IT, right? Within IT, your quality assurance unit probably reports to your IT top management, okay? This is the same. You need to think about within your biocontainment laboratory, your quality assurance people who are providing oversight, how can they report to ensure that any quality issues appropriately go up the chain of command? So the final question, and this is what we're really going to focus on, is how do you go about implementing this? So if you have your established unit, you know what their scope is, you know what their purpose is, and you found a nice little place in your organization to put them, how do you implement the processes into your system? So our recommendation is a continuous improvement initiative. And a continuous improvement initiative, this is the most standard quality assurance cycle you're going to see anywhere. It's the plan, do, check, act cycle, right? It's focused on looking at your system as a whole, identifying areas of risk, and then implementing quality assurance in a manner which addresses those highest level risks. So what that means is you don't have a checklist approach, right? You don't say, here's my list of regulations. Is the lab doing A, B, C, D? Yes, no, go. What you look at is what's the lab doing, or what's the space doing, what's the unit doing? Is it something which is resulting in a level of risk? If the answer is yes, what can we do to improve that process and reduce the risk? And that risk isn't always a safety risk. That could be a cost risk. That could be a quality of data risk. What you're doing is you're assessing and you're implementing quality assurance processes in a way that ensures that you have this ongoing continual improvement process. So I've given an example of what a quality assurance program that's set up for a biocontainment system might look at. So there, obviously, you'll notice a lot of things that Tammy mentioned in her presentation, such as SOP development, document control. A lot of those systems can be centralized through a QA unit. You can use, for example, the quality assurance should be overseeing to make sure that any training proficiency programs are actually working the way that you want them to, and looking at all sorts of facets of facility management, such as equipment management systems, and we'll talk about these a little bit in a moment. So the way we're going to talk through this and how to build a good, strong quality backbone is by going through the plan, do, check, act cycle, which is exactly what my officers would do when they're working with a lab or with a system and trying to implement a quality assurance process. So obviously you start in the planning phase. And as I mentioned, the planning phase is incredibly focused on risk. So you look at the system that you're working with and you analyze what the risks are in the system. So this is familiar. You've seen something very similar a few slides back from Tammy. So here you're performing conventional risk assessment, but when you're just choosing what your risks are, you're not just looking at your safety risks. So for example, if you're assessing your biorisk management system, your focus might be on safety risks, but it might also be on risks to reporting. It might be on risks that may occur with near misses, etc. So you'll generally create a risk register for any group that you're working with or any process that you're working with, and you're going to give yourself qualitative assessments of what the individual risks are. And by doing this, as you build out quality within your system, you can do it in a manner which addresses the area where you have highest risk. And that ensures that as you move forward, you're really hitting those areas that are most likely to result in a safety issue, that are most likely to result in an issue with the integrity of the data that you're generating. And by doing this, most importantly, if you're implementing quality where you don't have a requirement to hit a regulatory standard, you are most efficiently using your resources. Because by identifying those areas of highest risk and focusing on the areas of the highest risk, you don't have to worry so much. Oh, sorry, something just happened to my screen. Can you still see it? No? I'm so sorry. Can you see it now? Okay, awesome, sorry. So by focusing on those areas of risk that are highest, then you have the ability to move forward in the best way relative to cost. So once you've identified your areas that you want to address and you've identified the areas that you really want to focus on, this is where you get into the do part of quality assurance. And this is what you think of when you think of quality assurance. So I'm going to go through these slides a little bit quickly. But you start thinking about SOPs, right? Your quality assurance unit should be establishing the quality practices that other people are conforming with. And this is something that I have to remind our groups that we work with quite a lot. We are not the subject matter experts when it comes to a particular assay or when it comes to a biosafety practice. We're the experts on quality. So when we establish our policies, we establish the system within which your SOP should fit. So what that means is your quality assurance unit should be looking to have SOPs on things such as how internal auditing is performed, supplier management, something which is really, really important to ensure the integrity of a lot of what you do within the BSL3 environment, materials management, what's your chain of custody for samples, what's your chain of custody for reagents, etc. All of those things should be outlined in a quality system, but they should be outlined in a way that allows the labs to work through their own individual systems, right? You don't want to constrain what people can do. So the other thing I want to emphasize for your quality assurance system is they should establish their own SOPs as well. They should establish how they're going to function, how they're going to operate, much in the same way your biosafety unit would establish their own SOPs. Your quality assurance group should oversee your training programs. That doesn't mean that they're training people, except on quality, but they're making sure that the training programs that you have within your biocontainment program are rigorous and they're meeting the standards that you're looking for. So ensuring that when we develop training programs, right, it's very easy to make, okay, this is how we're going to do our training. You're going to read the SOP and then you're going to sign a piece of paper, right? That's a really good way to hit a requirement to say that people are showing that they're trained. But does that mean that a person's actually able to perform that SOP well? Are they able to do it effectively? How are you establishing proficiency and ongoing education? Document management, probably the most painful, but also the most important part of a QA system. So having a quality management system does allow centralization of all of your controlled documents. Basically, that group becomes responsible for making sure that everything is appropriately tracked, that you have version history, version control, and they also allow you to have some, Tammy was talking about security and other features. By having a centralized document management system through a QA group, you can make sure that all of your versioning and history is done appropriately. And it's also a great way to have a second set of eyes looking at documents before they go out. Materials management, another aspect of QA which is really important, is to ensure that you know what materials are tracked, what materials you need to maintain under certain standards. Your QA group should be overseeing requirements for temperature monitoring, for control of different departmental resources. But again, I want to emphasize you're doing this in a risk-based manner, right? So for your BSL3 labs, your supplies might be absolutely critical, whether your PPE is of high quality, whether it's providing the protection you expect it to might mean that for your biocontainment labs, your supplies are managed at a really high level, right? When you look at other labs that you in your institution, you might not be looking at supplies in the same way because maybe it's not a PPE concern, maybe it's a sample quality concern, a sterility concern, etc. And then one of everyone's least favorite groups, equipment management systems. So anywhere that you work, anywhere that you're doing lab work, you have to make sure that your equipment is functioning the way that it's meant to function. And the problem is that often there aren't resources to do it because equipment management systems are expensive. It's expensive to have someone regularly calibrate all of your equipment. It's expensive to have temperature tracking. So again, back to my favorite phrase here, risk-based. You want to look at what you're doing and look at what your areas of risk are. So for example, routine and regular calibration of your biosafety cabinets is not just high risk, it's required, right? So those are areas where you're going to put resources. But maybe there are other pieces of equipment that aren't required to be calibrated, but that provide a very high level of risk. So a really good example for me on this one is centrifuges. So your rotor cups, right, in the BSL2 laboratory may not be as critical as it is in a BSL3 laboratory where they're providing they're working as a containment device as well. So looking at all of your equipment in a manner which is risk-based is absolutely critical to having an effective quality management system. And then moving on to the check part of this. So there's no point in implementing all these things unless you check how well they're working. I can tell you that QA likes documentation, and sometimes we go in and we actually see instances where there's over documentation. We may look at something and we might see that you could actually do with streamlining things. So whenever you're implementing a quality management system, you have to make sure that you're reviewing those. So in order to effectively review the particular area that you're looking at, not all of these apply to a biocontainment program, you need to look at the system that you're reviewing, identify what your metrics are. So for example, if you're looking at a biocontainment lab and you're looking specifically at the biocontainment practices, you'll be looking at your health and safety metrics, right? You'll be looking at your near misses and your incidents. Whereas if you're looking at the quality of the data that's generated by the lab, you'll be much more interested in the rate of study deviations or their performance indicators for success. And that leads us into our internal audit process. So internal audits are absolutely critical for any quality assurance program. I want to emphasize the word audit is scary. So your quality assurance audit should always be performed in a non-punitive environment. What that basically means is I would recommend that internal audits are internal documents. They are confidential internal documents that are used to improve and enhance. They are not punitive. And these audits should be done in a manner which allows people to really review and properly issue appropriate corrective and preventative actions. And one of the things that I get asked 1,000 times is that if QAU audits all other programs, so if they're auditing biosafety and they're auditing the labs, who audits the QAU, right? It's the cycle of quality assurance. The answer is that your facility management, and this goes back to the scope and purpose of the program, should have checks and balances in place for the QAU. You can also have external companies audit your QAU. So if you actually have established regulatory standards, if you're working under ISO or GLP, there are plenty of people who come in and do that. And you have external consultants or sponsors will often come in and audit your QAU. So if we have a study, the external sponsor will generally come in and love to look through all our QA documents and make sure that we're functioning the way they want us to. And then also through TUBI, we will provide QA consulting services for other institutions. So, factoring time, I've got 2 minutes left. I'll get through it as quickly as possible. I know we're a little behind. So when you're looking at quality assurance, obviously you're going to have issues with conformances. So your quality assurance group should work out how to categorize their non-conformances, but keep that non-conformance categorization simple. You don't want more than about 2 to 3 categories, or it becomes confusing for the labs, for the groups who are using those. So an example here, which is actually the one we use, is basically to categorize our non-conformances into 2 categories. So I know it shows 3 here, but it's 2. We have minor and major non-conformances, and then we also, which is basically, minor is very easy to identify. They're low risk, they don't cause much harm, Your majors are causing harm, they're causing risk. The only difference between our major and critical nonconformances is that if there's critical nonconformance, it's leading to ongoing damage, and there's some requirement to immediately stop work. So these would be the instances where your QA people go into the lab and they identify a huge issue that they have to pass on to a biosafety officer, and the work has to stop immediately. But making sure that within your labs you have a clear reporting structure is absolutely critical. So your QA team should be formally trained in root cause analysis. They should be able to independently investigate those nonconformances. And what that means is that you're removing the bias, right? This goes back to my earlier slides about independent reporting. Your QA people should not be biased when they're looking at these incidents. They didn't design the biorisk management system. They didn't design the systems that they're auditing. So that helps them identify not your basic causes that are having an impact, but your true root causes of the issues in your system. So the final part of that Plan, Do, Check, Act is obviously the act. And your QA group should have a mechanism via which they can issue corrective action requests. And their corrective action requests should always be driven at resolving and preventing future nonconformances. This should not be a system which doesn't have a back and forth. It should always be done with the stakeholder in mind to ensure that anything that you're asking for is reasonable, that you're not over documenting, that you're not overburdening. And then your quality assurance group should take responsibility for re-inspecting after they've issued those actions to ensure that they are effective. I will note that one of the biggest challenges, especially if your QA group's working in an unregulated environment, is to make sure that they have the top management institutional support when they do have issues to ensure that those corrective actions are actually followed. And then my final slide here, continual improvement, right? So we have a cyclical approach, which means that we have to have ongoing improvement. And this shouldn't just be for your biorisk management system or for your labs. This should be for everyone. So the QAU itself should always do continual self-assessment and make sure that that there aren't ways in which they can improve, which I guarantee you there are. But also looking at reviewing SOPs on a regular basis, optimizing protocols, ensuring that you have that ongoing testing of proficiency, etc., all absolutely critical parts of having a functional and effective quality management system. So with that, I see a lot of questions in chat, so let me catch up real quick.
Okay, CARYS, I just wanted to do a quick sound check. If everybody can hear me a little bit better, let me know.
Loud and clear.
Perfect. We did have a 5-minute break built in after your Q&A session, but since we do have a lot of questions, if everybody is okay with pushing through that break, we can go ahead and get to these. So we'll go ahead and start with the first one. So CARYS, when you talk about quality management systems, are you also considering or have considered
Yes.
implementation of ISO?
So when we built out our quality management system, we started based upon GLP guidelines. And then as things have progressed, we've built in different systems. We are not officially regulated, though. We have not sought ISO regulation of our system simply because it's a cost factor to bring in an external company to regulate you. And unless you have a requirement for it, it's not necessarily seen as a benefit. But we do build in standards from ISO 9001. It was actually a requirement as part of the poliovirus essential facility. So we went back and reworked a lot of our SOPs to bring in those ISO standards in addition to the FDA standards.
And as follow-up, does Tulane already have in place an ISO 9001 system in place?
As I said, our systems are built on that, but we're not officially accredited to that standard. Performance metrics for the BSL3 labs. So when you're looking at, it depends what areas of the BSL3 labs you're looking at, right? Because your performance metrics, are you talking specifically about lab personnel? Are you talking about the biorisk management system? Because the metrics will change depending upon what system you're assessing. Compliance. So this is a newer part for us, right? Because historically our quality assurance program did not audit the biorisk management system. It is now doing it for the poliovirus program. And what we're doing, the metrics that we are looking at are near misses, incidents, the kind of standard metrics that you would look at, but then also follow up as well, ensuring that trainings are being done. We also have to look at, for specifically the poliovirus facility, we're also looking at things such as decontamination processes, validation processes, making sure that they're well documented. All of those factors have to be reviewed by QA for that system. But it is a little bit newer for us within the last 2 years, Henry. I think that's probably accurate to say. And then who's responsible for updates to the documents? So every single department is responsible for their own updates, but they all run through QAU. So if you have, for example, a PI wants to update an assay, then they'll make those updates. They're responsible for the technical content of the document, because obviously we cannot be biosafety experts. We can assay experts and experts in everything. What we're reviewing is from the document control point of view, from whether the documentation is sufficient for reproducibility of the procedure, whether there are appropriate training requirements within them, whether there's appropriate documentation within it. Audits, inspections at your institution, scheduled or unscheduled. I'll let one of Tammy or Anne-Marie speak from the biosafety side. From the quality assurance side, we do quarterly inspections for every lab that we work with. And then whether they're scheduled or unscheduled, this might be a sore point for the labs. We used to only do scheduled inspections. We have recently switched to doing all unscheduled inspections. And that's because we found that when we do unscheduled inspections, we get a much higher rate of non-conformances. So they are a far more accurate representation of what's going on within the system. We will occasionally do scheduled inspections if we have a group where we have maybe a lower concern. But no, we like to do unscheduled. It doesn't always make us popular, but it has been a lot more effective for us. And then finally, with risk assessments, I'm not sure about biosafety, but within QA, no, we just work in-house. We use a standard qualitative risk assessment matrix and document everything within a risk register. We don't have a tool that we use. Do you want to add anything from the biosafety side?
Yeah, so I'll just kind of echo, essentially, that's basically what we do. So for the actual safety inspections of the labs, those are annual, or whenever they submit a change or a new agent, and then we re-inspect, and those are scheduled. But then we do unannounced inspections when we have noticed or have gotten reports from other people nearby that there's been a complaint or something, where we're actually going up to follow up without giving them the heads up. And you know, it's not meant to scare them or blindside them. It is meant to catch what basically the same thing that ends up happening with QA is to catch what's really going on.
Okay, I think that rounds out our questions for QA. We can go ahead and next we'll have Nadia Golden. She will be presenting on creating a business model for BSL3 laboratories, and I will give her the floor.
All right, hi everyone. I hope you guys can hear me. Welcome to your crash course on creating a business model for the BSL3 laboratory. I'm Nadia Golden. I'm the Assistant Director of Research Administration. I manage the BSL3 laboratories as well as the High Containment Research Performance Core at Tulane University. Over the next 20 minutes, I'll walk you through the essential components of creating and managing a sustainable BSL3 lab from a project management perspective. So I do apologize. I'm a little under the weather, so my voice isn't there, but I'm trying my best to speak as loud as I can. All right. So BSL3s are specialized facilities crucial for infectious disease research, requiring specialized containment measures, safety protocols. You know this. As you embark on managing such a facility, understanding both the scientific and business aspects is essential. And so today we'll focus on turning your more technical knowledge into sustainable business operation. Oh, did There we go. All right. So obviously, managing BSL3 comes with its own unique challenges. The high safety standards and regulatory requirements add an extra complexity to every process that we do. Successful BSL3 labs thrive on robust risk mitigation strategies and stringent safety protocols. And so a lot of what I'm covering, you may have heard already from TAMMY, CARYS, and you may hear it again from Anne-Marie. We work so closely together that there's a lot of overlap in the work that we do. And so my team is very much connected through biosafety and quality assurance. So our primary responsibility as a manager is to balance these safety requirements with related research objectives and financial stability. So when implementing risk mitigation in your lab, develop a comprehensive assessment framework specific to your research focus. Each project should undergo thorough evaluation before approval, with clearly documented safety protocols that are consistently reviewed and updated. So I'm going to talk a little bit about establishing a core facility model. So one of the most effective approaches to BSL3 lab management is a core facility model. This centralizes the resources, enhances efficiency, and provides a framework for stability. So the high containment research performance core that I manage, we offer, well, it's an excellent real-world example of this model in action. We've created a comprehensive service that demonstrates how core facility should function. So our lab provides full or partial support to investigators conducting BSL3 research or select agent studies. Our centralized approach includes running assays on specialized equipment like Olink, mesoscale, PCR systems for nucleic quantification. What really makes this model effective is the fact that we standardize these processes. So the core standard operating procedures have been thoroughly tested to ensure consistent practices and have undergone rigorous quality assurance to guarantee a reducibility of all those activities. And this standardization is the hallmark for a successful core. It helps, investigators that are looking to perform BSL3 work, it takes the guesswork out of it. It allows them to reach out to a team that has been thoroughly vetted, that has already gained the experience, so that they are not having to take personnel in, they don't have to spend time training, it's there. So it's a very cost-effective model. So our service portfolio encompasses everything from processing services like bacterial, viral propagation, we do blood processing, to specialized BSL3 services, including rodent processing, single cell. And then in addition, we also provide a project management assistance for study design, budgeting, and regulatory preparation. So from the business perspective, this core model is really remarkably cost effective. By pooling those specialized equipment and expertise, the core creates a scale that makes advanced BSL research much more accessible to a broader range of researchers. And this maximizes the efficiency while significantly reducing the operational expenses that would have been incurred, like I mentioned, from having a research team go in and have to train to do their own BSL3 capabilities. So when developing your BSL3 model, consider how you might implement similar centralized approaches that optimize resource utilization while maintaining the highest safety of standard and quality. All right, so let's talk a little bit about the business plan and essentials. So a solid business plan is the foundation of your BSL3 operation. I cannot stress that enough, guys. You start by identifying your target market. Will you be serving academic researchers, pharmaceutical companies, government agencies, or maybe a combination of both? So each of these client segments have different needs and different funding mechanisms. You want to develop clear pricing strategies that account for all your operational costs while maintaining competitive rates. You want to consider tiered pricing models based on the resource utilization, the project duration, the technical complexities. your revenue projections should be realistic and account for facility, maintenance, certification, staffing, and equipment replacement costs. I apologize if I'm going through this too fast. All right, so let's also reach out and think about operational considerations. So staffing is a big one. You know, your staffing and equipment management, they're crucial operation components. So determine your staffing needs, which are based on anticipated workload and required expertise. Every team member needs specialized training in BSL3 protocols, which represents a significant investment. Budget accordingly for the ongoing training. Our personnel spend anywhere from 3, sometimes up to 6 months training before we even let them in the BSL3 by themselves. So, you know, we go through tons of proficiency training. They're paired with a mentor, a senior personnel in the lab that goes in, watches them, and then we go in and observe as well. And we also have a proficiency checklist that is provided by biosafety that they have to go through before they get checked off to go in by themselves. Also, equipment management in the BSL3 environment extends really beyond procurement. You need to establish rigorous maintenance schedules, certification processes, create systems that ensure continuous monitoring of critical equipment and containment features. And remember, documentation is just as important as the procedures themselves. And I know QA stresses that a lot. So I am going to touch briefly on QA. I know CARYS really went into depth, but again, I feel like if we're mentioning twice, it's probably worth noting. It's you know, it's important. QA is such a large part of our processes. So implementing comprehensive quality control measures is non-negotiable for BSL3 operations. Your quality management forms the foundation for both the safety compliance and the operational excellence. So this system should include documented quality policies that align with institutional standards. It should clearly define roles, responsibilities for quality oversight, structured processes for identifying and addressing nonconformities, regular internal audits with formal follow-up mechanisms, management review processes, training requirements and assessments. As I mentioned, in our lab, we do proficiency training on processes, processing techniques and assays. And then also document control procedures for SOPs and protocols. Also developing key performance indicators. So develop your key performance indicators that track not only the research outputs, but the safety compliance, the operational efficiency, and the financial performance. These matrices should include the safety compliance rates, incident and near miss frequency and resolution matrices, equipment downtime and maintenance compliance, the turnaround time for sample processing and results, the cost per sample or service unit, resource utilization rates like staff time, equipment usage, client satisfaction scores, and feedback matrices. So maybe consider sending a post survey for feedback to your users to help improve your processes. See what's working well and needs improvement. And then lastly, your recharge rate accuracy, your projected versus your actual revenue. And then lastly, continuous improvement mechanisms. Your quality management system should include regular audits, corrective action plans, and continuous improvement mechanisms. So implementing formal corrective and preventative action processes, root cause analysis, change management procedures, regular quality meetings with documented action. So if you do have a quality assurance program on site, it would be good to rope them in at that point. You know, have them be a part of your processes. If not, there are things that you can do internally as well to kind of create your own quality program. Also benchmarking against similar BSL3 facilities, process mapping to identify inefficiencies, have staff involvement in improvement initiatives, and technology assessment for potential enhancements. And so this systemic approach helps maintain high standards while identifying opportunities for operational enhancements. It also provides the documentation needed for regulatory compliance and demonstrates the value to the internal leadership and your external stakeholders. So let's talk about marketing and partnerships, because even the best BSL3 facilities, we need clients. We need people to use our services to be sustainable. So develop a targeted market strategy that effectively communicates with the value proposition to your potential users. Focus on unique capabilities, expertise, and your track record of safety. Strategic partnerships can significantly enhance your sustainability. So explore collaboration opportunities with academic institutions, industry partners, maybe government agencies. These relationships often lead to more stable funding streams and can help expand your research opportunities. So, for example, my lab's recently, we've recently started running O-Link assay. We're one of the few, we're actually the only facility in the South that runs O-Link, and one of very few in the US. And so we're starting to have pharmaceutical companies, other research facilities reach out to us because of this technology. So find something that puts you above the rest, that really draws attention to your facility. And so with that, I'm going to move into recharge rates. I don't know how familiar you guys are with recharge rates. But so I'll try to break it down as best as I can. So the financial management of specialized research facilities like the BSL3 labs requires a systematic approach to cost recovery. And so this guide introduces that fundamental concept and practical steps for developing a research rate model that ensures both regulatory compliance and financial suitability. So what is a recharge rate? At its core, the recharge rate system is a financial mechanism that allows specialized research facilities to recover the actual cost of providing services. So unlike commercial enterprises that aim to generate profit, the research rate operates on a break-even model where the users pay for the direct cost only associated with the services they receive. So this system is distinct from indirect costs, your IDC, also known as facilities and administratives. The IDC is the overhead expense that supports your institutional-wide operations, such as the building maintenance, utilities, administrative functions. So when researchers receive grants, a percent is automatically taken as IDC to cover those broader institutional costs, which is why the recharge rate system focuses exclusively on the direct cost system. So there's a simple formula. The recharge rate model rests on the fundamental equation, the annual cost divided by the annual usage, and that gives you your break-even rate. So this straightforward calculation ensures that all direct costs associated with providing the service are fairly distributed among users based on their actual utilization. For example, if your BSL3 lab costs $100,000 to operate annually, and you process 1,000 samples, your basic rate would be $100 per sample. So let's talk about creating your BSL3 recharge rate model. So developing an effective recharge rate model involves 5 essential steps. 1, define your service clearly. Creating a sustainable recharge model begins with precisely defining what you're selling. Consider how your services align with resource utilization and user needs. This includes determining appropriate units of measure, samples, hours, or procedures, deciding whether to bundle or unbundle. We give those bundling, unbundled deals, like you see on Verizon or AT&T. We have bundle, unbundled deals. So that includes kits without kits. Including flexible options like adding consultation rates or non-standard requests. For BSL3 facilities, this might mean charging separately for lab access time. Could be sample processing, specialized procedures like viral culturing, or equipment usage. You can even charge for training. You can charge for a usage fee for someone to use a room. So there's so many different ways to get that recharge rate through utilization of your BSL3 core or facility, sorry. So calculating your direct costs, a comprehensive accounting of all allowable costs forms the foundation of the accurate rates. This includes annual totals for personnel salaries and benefits proportional to the time spent on the service. Lab supplies, consumables, service contracts and maintenance, equipment depreciation, which only applies with if purchased with non-federal funds. It's critical to capture the total annual expenditure rather than just the ideal cost per unit. This approach accounts for necessary expenses that aren't directly billable, such as preparation time, failed runs, and administrative tasks. Excuse me. Number 3, allocation of costs appropriately. For facilities offering multiple services, costs must be distributed across categories based on actual utilization patterns. This process involves assigning expenses to services based on percentage of use, tracking staff time, allocation across the different activities, ensuring complete distribution of all costs. This allocation method acknowledges that resources like staff time, equipment are often shared across multiple service types, and costs should be distributed accordingly. And let's see, project realistic usage levels. Accurate projection of service utilization is essential for setting appropriate rates. So this involves analyzing historical usage data when available, communicating with current and potential users about upcoming needs, understanding the capability limitations of your facility, and accounting for funding cycles and research trends. Overestimating usage leads to unsustainable rates and potential deficits, while underestimating can result in excessive charges and compliance issues. And then lastly, number 5, calculate the rates and apply necessary adjustments. The final step involves applying the basic formula and incorporating appropriate adjustments. So dividing that allocated cost by projected usage for each service. Adjust for user categories, internal versus external versus commercial and industry. Account for any previous surplus or deficits. Consider subsidies if applicable. So any of your costs may be covered, like PPE covered by the facility. So you might be able to provide users with discounted rates in that instance. For external users, rates typically include additional components. So academic institutions pay the eternal direct cost rate plus the IDC rate, while commercial or industries will often pay market rate, which includes the direct cost rate with the IDC rate included into that base rate. So at our facility, when we're working with outside collaborators that are non-NIH funded, we will take our base rate, tack on the 75% IDC, and that becomes the new base rate. And then our facility will also add a 30% administrative fee. Versus if it's an NIH funded, it'll just be the base direct cost, and then they'll pay IDC on the back end. Excuse me. So financial management best practices, so maintaining compliance, financial stability requires ongoing attention. We actually check our recharge rates on an annual basis. So every year we will go through, whether it's Fisher's website or Sigma, whichever resources, to ensure that we are always up to date on our recharge rates. So we are not overestimating or underestimating, that we are pulling the most current rates available on the market. So maintaining compliance, regular reviews, consistently invoicing, billing users on regular intervals, detailed documentation, so keeping comprehensive logs of the facility usage. You want to separate your track and monitor different fund balance components like your operations, your depreciation, your external F&As. Some common pitfalls to avoid: failing to adjust the rates to absorb previous surplus or deficits, basing calculations on idealized rather than actual costs. Because remember, we're working on a break-even model. We're not trying to make a profit out of this, unless you are. Using surplus funds for unrelated activities, applying inconsistent billing practices across user groups, including costs already covered by the IDC. By understanding these fundamental practices and following established best practices, BSL3 lab managers can develop recharge rate models that comply with the regulatory requirements that ensure long-term financial stability of these critical resources. So I'm going to give you guys an example. I apologize. So this is what our recharge rates look like. We have a recharge rate for everything, and I mean everything. So this is what our recharge rate be for processing a 7.5 ml plasma serum tube and our BSL3 PPE. We take each and every item that is utilized in that instance, the amount used, how much is in a pack, the percent of the unit used, and that gives us our price that we charge. So when we go into the lab, we are charging you for the tubes that you're using, the PPE, setting up the biosafety cabinet, the disinfectants that we're using, the freezer boxes. We charge for, individual storing, because keep in mind, when they're running studies in BSL3, those samples cannot come out unless they're inactivated. So they are utilizing space in your freezers, and you're paying for that. So create a recharge rate that recoups that cost and helps pay for the maintenance of your freezers. And then lastly, I just wanted to touch on some project management tools that we use. So effective management of the BSL3 lab requires robust systems to track workflow, ensure compliance, and maintenance of financial oversight. And so the following tools can significantly enhance operational efficiencies and project management within high containment facilities. So we just recently came on board with iLabs. iLabs, it's been great. So it's a way for, if you have services that you offer, or maybe you have multi-user rooms, it's a way for others to schedule a room for use or submit a request for a service or assay to you, and you can then in turn submit your billing. You can let them know the process, rather than them emailing you 5-6 times. When is my study starting? You'll communicate through this platform, and it'll say, okay, well, it's in review with KENWAY right now. It's in review with biosafety. Now the study starts. So it's been a really helpful tool. My team's really enjoyed using iLabs. We've tried Monday and Asana. Personally, I like Monday more than Asana, but both give very similar features. It's a project management software tool. So if you have multiple users or multiple technicians in your lab, you can assign them tasks through Monday. It's very interactive and colorful, and I think that's why my team likes it. So when you're done with a task, it gives you like this little, like, huzzah, you did your job. So they really enjoy using Monday. It's a fun tool. I think it's free if you wanna. They do have subscriptions, I guess, based on your need. Lab Archives is also an electronic notebook. This one's been highly suggested through NIH. They've really been pushing Lab Archives, but as we kind of move away from writing in our notebooks, Lab Archives is an online research notebook. So you can post notes. My team mainly uses it for our quality assurance documentation. So they can, as they're processing, they can go onto Lab Archives while they're processing and check off on the different steps that they're performing. I know others at our facility uses Lab Archives for scheduling necropsy samples. Sorry. They use it for scheduling necropsy and thanks. So as a scheduling tracker, I believe Lab Archives also gives an option for inventory management, although we have a different system that we use for inventory management, but they do offer that as well. And I think that was my last slide. All right.
Great, thank you, Nadia. We have quite a few questions for project management as well.
Oh, no.
With recharge rates, but Alicia did ask, how do you determine if your services are competitively priced? And this has actually been difficult for her because they don't want to be viewed as attempting to find this information in any disrespectful way. So I know that we're kind of different with our break-even rate, but with those external clients, how do you assess the cost on there?
That can be really hard. You know, I'll be honest, I've looked on other people's websites, like I'll go to Duke University and other BSL3 facilities. A lot of them will have their costs listed on their websites. I mean, sometimes you'll be competitive, sometimes you know, it's really hard to gauge because at our facility, we do have set rates. We have to follow a 3% increase on our costs each year. And each region is going to be different too. Depending on where you're located, your costs would look different than someone in the South just based on our operation needs. So I would just, you know, do some digging online, see if you can reach out to other BSL3 facilities. Most of them are more than happy to share their recharge rate with you. I'm more than happy to share mine as well if you want to reach out to me. You know, but most facilities are very open to letting you know about their charges.
Okay, great. Okay. And the next couple of questions we can go through. So there's a question regarding, is there a centralized approach that includes provision of PPE to PIs? I think that we may have gone over this with your slide discussing everything, but I'll let you go a little deeper into that. Also, what are the costs associated with these services, such as space, salary, facilities, electricity and water, and any operating and maintenance activities performed in-house?
Just gonna make sure I cover everything.
I know that was a lot.
Yeah. Okay, let's see. Centralized approach includes provision of PPEs. Yes, yes, it does. So we have a rate for PPE that we charge a rate for utilizing the cabinet. So anytime we go in and perform a study, we are charging for your PPE, and that includes, you know, your gown, your gloves, your sleeve covers, your booties, all of that is included in that rate. What are the costs associated with the services? So things like facility services, electricity, water, that's going to be covered in your IDC. So we do not cover that. But we will cover things like your equipment maintenance. So if you've got centrifuges, freezers, your different specialty equipment, all of that is included in recharge rates. And we may set that up as a user fee. So we charge a one-time user fee, and that covers the project management aspect of me doing your iCook, your IBC, helping with your schedule design, the labeling that it takes my team to label the 5,000 tubes you've now requested, the maintenance of some of the specialized equipment. So it is a specialized fee, and it is based on the user. So we do a lot of custom charge. You will find that by setting up the core model, you're going to do a lot of custom charges. Some things will stay the same, like blood processing, necropsy processing. But when you get to those specialized assays, they're custom rates.
Okay, and I think that also covers the question regarding projects that might cost a little more because they have more things. Okay, and are there any activities that, according to Tulane's research administration process, will not, like the administrative requirements won't allow for cost? Maybe pathogens or other considerations that may not be included?
I haven't run into any issues. Again, our rates are custom based on the investigator's needs. So at our facility, we do tuberculosis, COVID, we have select agents. So each time an investigator reaches out to us, if we don't have a rate on file, we'll create one for them specific to that agent. But it still includes PPE, setting up a cabinet, all those things, because we need to get that money back.
Okay, and I like the wording on this one. Do you offer any of this gorgeous QC or cost documentation to clients to reinforce confidence in the services, product, or minute spaces?
I have not, but definitely something to consider.
Okay, and Alicia, if you would like to elaborate on that question, that is perfectly fine for you to come off mute.
I can. I do want to add a little bit to the previous question because it kind of overlapped with another one asking about the DIRC policies that are updating as well.
I missed that one.
So when I read the question about the RA process, I thought it was risk assessment process. So I will say that one thing that we are definitely doing very closely together, when Nadia mentioned how close we work together, is one of the overlapping roles with biosafety is referring back, is when she gets a project submission, she's checking with our biosafety group to see if they have their IBC approvals in place. And then we're also involved in the initial planning discussions to find out if this is even something they can do. So for that, referring back to that risk assessment question, is this even something that we would allow at Tulane? We start to have that discussion before they even get to the point of submitting it through the IBC to maybe save them a few steps as well. So that's kind of built into this collaboration between these units. And then as far as, incorporating the DIRC, our institution is considering making it part of the IBC process, so they won't have a second step to go through. So that'll be part of the regulatory check.
CARYS, I don't know if you maybe want to touch on QC documentation, anything to that effect.
I mean, so I'm just going to say all of our QA documents are internal documents. We don't share anything with anyone else. There are a couple of very, very rare exceptions. If we have a situation where we have maybe a study deviation and we have a sponsor who is looking to see that we have a good and effective corrective follow-up to that, we may share a statement, but generally all of the other documents, as beautiful as they all are kept internally. So that's actually very deliberate, because it's one of the things that our programs really focus on is having a high level of integrity and trust between the QA unit, between any of the other groups that they're working with. And by keeping all of our documentation confidential, I think that it allows a huge amount of honesty and trust to exist within the organization to ensure that things are handled in the best way. So we do provide, there are things we do provide externally. We do provide records if they want to see how regularly we're performing audits, if they want to see when we're performing QC checks, schedules of those kind of things we will provide. But actual documents generally stay internal.
Okay, thank you, everyone. I think now we are going to move on to Anne-Marie May. She is our Executive Director and will be presenting on biocontainment operations.
Thank you, Haley. So as she mentioned, my name is Anne-Marie May, and today we'll be talking about ensuring safety and compliance through biocontainment operations. Can everyone hear me? Perfect. So what I'm going to be talking about is going to pretty much relay exactly what everyone has been talking about this whole session, because I think it's really important to identify how we all work together to maintain the safety and compliance of our biocontainment operations. So first, what is biocontainment? It's the combination of physical parameters and operating practices that protect the personnel and the environment and the community from exposure to biological agents. So how does it work? It works through multi-measures to contain biohazards through your facility, equipment, techniques, and established practices and procedures. Biocontainment barriers are fundamental to the safety and function of biocontainment facilities. They use physical and procedural methods to isolate and contain pathogens to prevent their release and protect the personnel. Barrier protection is accomplished in 2 layers, primary and secondary, which is something that Tammy had touched on earlier in this talk. I'm going to deep dive a little bit more on each barrier, just because I am talking about the operations itself. We're talking about the facility. So primary barriers or the equipment are items within the laboratory that isolate and contain the biohazard. Use and function of these barriers is determined by standard operating procedures, SOPs, as we've all been talking about. These are very important. They shouldn't just be specific to the work that you're doing, the procedures, but also the equipment and the building and the safety and emergency response. You want to identify your equipment needs and how it functions, and you want to identify how are you going to decontaminate this equipment? How are you going to get between pathogen use if you're a multi-use facility with different agents, and then repairs and preventive maintenance. Not all facilities or contractors will be able to go into a high containment facility. So how are you maintaining that equipment or deconning it to the point where it can come out for repair? I am going a little fast just because I know we're running a little short on time, so I'm going to kind of push through and then we can answer questions at the end. Some examples of primary containment devices are biosafety cabinets, centrifuge rotor cups, lids, and PPE. And then also examples of equipment that you need in the BSL3 that you should really risk assess prior to going in. How are you going to use this equipment within your facility? Do you need to QC it, calibrate it? How is it going to function? Is it aerosol producing? Your secondary barriers is going to be your facility. It's a physical lab and it's designed for containment. You're going to have directional inward airflow that moves from clean to contaminated areas with approximately 10 to 15 air changes per hour with a differential room pressure of 0.05 and measuring in water gauge. You're going to have 2 self-closing doors with an ante-room entry where you can get dressed in your PPE. Restricted access and signage is important for this facility. All room penetration should be sealed and should be easily decontaminated. And exhaust air should be, HEPA filter should be considered, depending on the work that you're doing. And again, it's all about risk assessment. Biocontainment operations, procedures, and practices that isolate and contain potentially harmful organisms. So I'm now going to kind of go into the operations and establish procedures. Established practice and procedures, SOPs should be considered for PPE, entrance requirements, emergency response, equipment use and maintenance, and then experimental procedures. You should use a risk assessment process for your SOPs and identify, review, and control and evaluate, as we've been talking about throughout this presentation. Your facility manual should have these SOPs reviewed and updated as needed. It is very dependent. Some people might do it annually, some might do it more often. It's just based on any changes that may need to occur and your facility requirements. And then it's really important training on these practices and procedures are a critical component for biocontainment operations. I'm going to deep dive into a training program because I think it's really important for your operations is to have an established training program that works for your facility. You need to identify who needs to be trained. It's not just going to be your laboratory staff and faculty that are working in the laboratory. You're also looking at your facility workers. What about IT that needs to go into work on your computers or electrical equipment for you got emergency responders and security, occupational health and safety, contractors and visitors. What information does your group need to provide? Do you need to be provided? It's something you really need to consider. What information are you providing to each of these individuals? Is it the same information? Is it specialized? Is it hazard awareness? So as we know, training can be done in person or virtually. I think there's no one size fits all. It's based on what works for you. I think the most important thing is identifying how they understand the material once you do the presentation. So are you doing verbal confirmation of understanding? Are they signing a document of understanding or maybe even a test? I think it really depends on who you're training, what you're training for, and it really needs to fit your process and procedures. And then walkthroughs. One thing we do at Tulane is not only do we do the information-based sessions where you sit in a PowerPoint, as we say, you just gotta sit through that 4-hour training sometimes, and you have to get through it. But next after that, you're going to go through your walkthrough, and that's what we call our initial walkthrough, our first walkthrough. And we do that during a low hazard time when there's not a lot of work occurring in the space, so that we can allow those individuals comfort in putting on the PPE, walking through the space, asking their questions that they might have. And then they do proficiency-based training with a trainee, which is, again, as we talked about, is a senior person within the laboratory, and they go hand in hand with the trainer and the trainee to become proficient in the procedures that are specific to their work. And I do think it's important to note, we've been talking about lab-based people, facilities, IT. Your facilities people don't need to necessarily know how to set up a biosafety cabinet and an assay for blood processing, but they should definitely know how to put on PPE, what disinfectants are you going to be using in the facility, what risks are associated, what agents they're working with, and maybe some of the equipment. What are the hazards of some of the equipment in this space? Why is this space so important? And I think that's the big thing that I find with our facility workers is and visitors and contractors who don't understand biocontainment is, why is it so important? What's different about containment versus this office space that I just went to a few weeks ago? So consider that in your visitor training and facility trainings as well. So again, questions to consider for your training. How are you identifying proficiency in your established practice procedures? At Tulane, we do have a list of what we identify as proficiency for our lab staff. What are the requirements for entry, and who verifies who met those requirements? So we got the trainer and the trainee process. How often do you need to provide the training? Do you do it annually, refreshers, initially? What about we do kickoff meetings. So are you doing, I think you should consider doing agent assessments prior to the start of the study, as well as just refresher trainings for staff. So a point we always like to make is conversation does not equal confirmation of understanding of material. I wanted to share that with everyone here. Communication is really important. I think you need to identify your stakeholders. It's not only leadership, but also your facility staff are maintaining that function. So identify who are your stakeholders in your facility. Plan with them on the expectations, coordinate schedules, and provide a schedule of work so everyone's aware of what's occurring, not only in the laboratory, but facility-wise. Effective communication is crucial for a successful biocontainment program. I'm going to talk a little bit about preventive maintenance and calibration. So preventive maintenance is the practice of regularly maintaining equipment and systems, as well as calibration is adjusting the measurement of instruments to ensure the readings are accurate and reliable. Establishing a checklist of preventive maintenance and calibration is important for scheduling and coordination. That way you are aware of what equipment, and this is not specifically what's in the laboratory, but also your building PMs, your fans, air compressors and vacuums and autoclaves. They all require preventive maintenance and calibrations. So you want to make sure to have that scheduled together and know when it's going to occur, and make everyone aware within the laboratories that you might have to shut down that autoclave for a week just to get a PM and calibrated. Or a week, it might be over exaggerating, but just, you know, make sure they plan appropriately. Validation of facilities is the establishment of performance characteristics of a method and provision of objective evidence that the performance requirements for specified intended use are fulfilled. And I got this from the ISO standards. It's important that you validate equipment after PM and calibration is beneficial. Basically, it's beneficial in providing evidence of the systems. One thing that we do for like biological indicator validation for autoclaves, after you've gone through your PM and calibration, now you're retesting and re-verifying and validating your autoclave process and procedures. Facilities verification, it demonstrates the validated method functions and users and according to the method specifies and validation is fit for purpose. It's important that you identify what standards your facility needs to meet, and then risk assess what should be done and whether should your facility be shut down for this verification. Does it need to be? Maybe, maybe not. I think it really is important that you risk assess what you're doing during your verification processes, what's currently ongoing in your facility, and whether or not you need to completely shut down to make sure you verify your facility appropriately. Examples of facility verification items: building alarms, facility equipment, air change rates, differential pressures, failure scenarios. And these are just examples. There's a whole list more items that you can that you might need. So how often should you verify your facility in the biosafety world? We always say it depends, and I think it's appropriate here as well. Have you changed anything in your facility? Is your BAS system? Have you changed anything in your programming, upgraded your fans? What regulations or standards are you required to meet? And who does the equipment and facility or contract recommend? There might be recommendations based on your commissioning agent or your equipment manufacturer or facilities department that state that they how often they want it to be PM calibrated and verified for function. So just quick summary, BSL3 facility design and maintenance and verification of critical function equipment are high priorities. Establishing SOPs and training programs are essential, and coordination communication is vital for all successful biocontainment operations. I know I went through that really fast, but I feel like the questions that we've been getting have been really great. And I do want to keep it open to more questions for all of us. So yeah, that's it. That's all I have for today. Thank you.
Great, thank you, Anne-Marie. And I hope that all of you will go ahead and continue the conversation. Again, this training was kind of put in place to showcase how we have established these disciplines within our department, having dedicated QAU, having that dotted line connection to our program managers, just to kind of put us in a place where we are able to stand up operations and scale up or down, depending on what we're dealing with very easily. That collaboration and that access to everyone really has kind of created a seamless process for us to be able to take on new studies. Again, I came in during the pandemic, but watching that process and seeing all of these people that are on this panel respond to that in kind was a very, very impressive thing to watch. So we do encourage you to stay on and ask questions about how we do that. And I do see that we have a question from Louise that says, do an in-person training. So, okay, so we can see how everything works. Very interesting indeed. I will let you know that we do have the capacity to host groups in person here at Tulane. I'm pretty sure that we may be willing to travel as well. But we do have a 5-day program in place that kind of does a deeper dive into all of this, which includes tabletop exercises, hands-on training within one of our BSL3 locations, tours of our facilities, And it gives the opportunity to have that very immersive opportunity and experience on our campus to see how our team works and how they make the magic happen. So I would encourage you to reach out to us either through email, visit us on LinkedIn, and we can provide you with more information about those resources. In addition, we will have programming throughout the year virtually, and as CARYS mentioned in her presentation, we will be offering consulting through quality assurance. So if there's any interest in that, again, please get in touch with us. Okay, and another question, have you considered establishing a network of BSL3 labs, maybe with other institutions in preparation to respond to emergencies if needed, or just to provide guidance to other international folks that I'm sure will be very interested? So I will say that the expectation is that the Institute will have a global reach. That was one of the missions and original initiatives of our former executive director. And in her spirit and legacy, we absolutely plan to provide training opportunities outside of just what we're doing right now. And let's see, do you provide hands-on training for animal work within a CL3 environment? The answer to that is no. But Anne-Marie, I will go ahead and let you speak to that if there is any expectation that in the future that would be a possibility.
Yes, if you're specifically talking about hands-on training with animals, that's a little bit more complicated, I think, than to do a hands-on specifically through TV. But it's something that we can work with our veterinary medicine department to see if that's a possibility. But I appreciate the answer, the question, because it's something you can look into if you think that is interesting.
I did have a question for you, Allie, though. Are you asking this question because do you have an animal facility in your current institution that is containment? Because what we do here is we actually do that training with the animals at a lower level. They pretty much do the procedure, at least for the mice. Not so much for the primates are a different story because they have dedicated personnel in the Corps for that. But for our other institutions where we're training people to do those procedures in the BSL3 environment, we do it at a lower level. So they have the opportunity to wear the PPE if they want while they're doing the actual activity. But they're not doing it in containment, and they're not doing it with the virus or agent present.
Exactly, and just to kind of piggyback off of that, any of our hands-on activities that would take place through the Institute would be in our downtown tropical health and the public health building, and in a location that's fully deconned and does not have animals in the space, just to protect the staff or attendees, but to also give them that completely immersive experience of having that hands-on in the lab training with the PPE and everything, just to make it as realistic as possible. Okay, anyone else? We are at the 3 o'clock mark, so I know that we had this scheduled a few minutes past, but again, we encourage all of you to contact us if you are interested in additional opportunities through the Institute, whether they be virtual, in person. We are sending out a post-event survey, so we do ask that you provide feedback to us just to help guide where we go next as far as interest and topics. And then also, since you did attend today, you will have access to video on demand. So you'll be able to use that as reference to bring back to your facility, and hopefully this has been something that will enrich your programs, and we hope to see you again. So with that, I'll go ahead and close. If any of my associates have any closing words, I'll open the floor. Okay, no takers. Okay, well, thank you again. We'll go ahead and end here, and we'll hopefully see you soon. Thank you.